DNA replication. So here we have a strand of DNA unreplicated. Notice that the nitrogenous bases are labeled and adhere to base pairing rules, adenine to thymine and guanine to cytosine. Here are the 3' and the 5' ends. Now this DNA will be the parental strand in the process of DNA replication. The enzyme helicase comes in and separates the DNA at the replication fork by breaking the hydrogen bonds between the bases of the two strands. Then another enzyme, DNA polymerase, replicates the split DNA by synthesizing the strand with free nucleotides, following base pairing rules, of course. This will, with time, create another complete strand of DNA. Now, whether the 3' or 5' comes first out of the helicase matters because replication must be anti-parallel. Polymerase adds bases a certain way depending on the direction the 3' end is in. So this bottom strand, which is flowing in the opposite direction, will have to be synthesized differently. The two strands are called different things because of the way they are synthesized. The top one, whose polymerase moves towards the helicase, is called the leading strand. The bottom one, whose polymerase moves away, is called the lagging strand. On this lagging strand, the polymerase will create Okazaki fragments, which will need to be glued together later to complete the DNA strand. In this case, the enzyme ligase comes in. Ligase connects the Okazaki fragments created by the polymerase and completes the strand of DNA. Now we have two newly replicated strands of DNA. Oh, and one last thing. This replication process is often called semi-conservative because each of these strands has one piece of the original parental strand.